Well, hello everybody. I'm Arthur Brandwood, the founder and principal consultant of Brandwood Biomedical, and welcome to this month's Brandwood Live webinar. Today we're going to talk about biological evaluation. I'm very pleased to welcome my colleague James Morrison, who's going to present our webinar this morning. I'm going to talk on the subject of how the FDA and the ISO stars are aligning on ISO 10993 and what's happened with the recent revision of ISO 10993 Part 1, the primary biological evaluation standard. Before I begin, just a little bit of housekeeping. This is a live webinar, and we will take questions at the end. So if you have questions as we go through the webinar, please enter them in the question and answer tab of your webinar control panel. You'll see two little tabs there, one called Q&A and one called Chat. Use the Q&A button, not the chat, so we get all the questions in one place. We'll be able to see your questions as we go through, and at the end we'll do the Q&A. So without any further ado, I'd like to introduce your speaker for the day. And there we go. So I'm Arthur Brandwood. I've got a background in biomaterials originally, and I've been doing biocompatibility for rather too long. I think I'm about the second longest serving member of the ISO committee that writes the 10993 series of standards. I've been doing it since the early 1990s. Participated most recently in the drafting committee for the 5th edition of Part 1. And my background's in biomaterials and regulatory. The speaker today, James Morrison, is Senior Consultant at Brandwood Biomedical. He's been a member of TC194 also for some years, and he currently serves as the head of the Australian Technical Delegation. He's got a particular expertise in respiratory devices, and James was very active in the development of the new ISO 18562 series, which is a separate series of standards for doing biological evaluations and risk assessments of devices that have a breathing gas pathway where the exposure is via the gases. He's currently heavily engaged in the Part 17 and Part 18 revisions, and James is a materials scientist, a toxicology background, and I was delighted when he was able to join us as a senior consultant here at Brandwood Biomedical earlier this year. So without further ado, I'm going to hand over to James and welcome and let you make the presentation.
Thank you, Arthur, and welcome everybody. I'm glad to see that we have a list of participants that reflects a global interest in this subject. So welcome to our presentation this morning. At least it's morning here in Australia, and we trust that your day is going well wherever it is you are. So ISO 10993 Part 1 is the biological evaluation of medical devices and its evaluation and testing within a risk management process. And in August this year, the 5th edition was published, and that replaces the 4th edition, which was from 2009. So as you can see, that's about 9 years between versions of this very important keystone document in the biological evaluation of medical devices. 9 years is probably a long time to wait for a revision of a standard, but it's a pretty substantial standard, and revision does take a long time. Also, I know that the ISO Technical Committee does not rush into revising editions. Rather, they wait until there is a sensible body of work that is backed up by a body of knowledge and expertise, so that editions are published in a sensible and significant fashion. So it is published by ISO, but it's yet to be published at national levels. So every country will have to adopt it as its own standard. And so there is always a gap between publication at the ISO level and broad acceptance at national levels. However, there will be national acceptance because the ISO process is actually driven by participations from all around the world. And so we will see in the coming year a national acceptance of this new version coming through. You also should be well aware, I'm sure, that there is guidance from various authorities having jurisdiction And naturally, I'm referring to the US FDA and the China FDA and a number of other bodies of that nature. And they have their own guidance about biocompatibility, and I think we're all familiar with FDA in the US and their guidance on this document. Those guidances are still going to be the way that authorities having jurisdiction are treating this new document. Their guidance is still going to be the ruling thing. However, you will see from the new edition the shape that biological evaluation is going to take moving forward. And it's quite common for those authorities having jurisdiction to follow the published standard just by some time. So let me enumerate the changes. The principal change that everybody, I'm sure, is interested in is Table A.1. This is the centerpiece of the document, and this is the table that identifies endpoints to be addressed in a biological risk assessment. And the important thing to see here is that this time, this table is normative. So historically, the Annex A, where Table A.1 lives, has been informative. That is, it just speaks to the rest of the standard, but it's not enforceable. As a normative annex, the content of Table A.1 is now part of the enforceable standard. So if somebody wishes to claim that their medical device biological evaluation is compliant with this standard, then it will have to be compliant with Table A.1, and you will have to demonstrate that you have addressed the endpoints that are nominated in the table. This is a substantial change. In the past, the table had text in it which said to the effect, this is not a checklist. And while it still isn't a checklist per se of tests to be done, it certainly is now a checklist of endpoints to be addressed in a risk assessment. So that's a significant change, and I do hope that everybody is paying good attention here and realising that they are going to have to start itemising these endpoints and addressing them in the high-level documents about biological evaluation for every device. There are some new columns, and I will show these columns in the next slide. And those columns are for a number of different things. One of them is not a biological endpoint, but rather it's an analytical materials endpoint, if you like. And that's the physical and/or chemical information column. You'll also see that there is a column for degradation. This is an understanding that there are increasingly devices that are designed to degrade, and they need to be considered in particular. And so where devices are intentionally degradable, sometimes this is called absorbable, then they need to be dealt with. And these are endpoints to be considered, and they're marked with an E. They're not tests to be conducted. This means that when you see that you have to consider an endpoint, it needs to be shown in your final document that you have considered it and what you thought about it and what you did about it and what the outcome was. It doesn't necessarily mean that you have to conduct a biological test. There has historically been an attitude of just doing the checklist of biological tests. Clearly, in these more enlightened times when we are rightly concerned with animal welfare, we are attempting to reduce the number of tests that involve animals and the number of animals involved in those tests. So the standard is explicitly written, not around testing, but around endpoint consideration. Let me now show you a cut from the Table A.1. and you can see that physical and chemical information is in fact the first column in the endpoints after contact duration on this table. And there are X's for every contact duration and every body contact type. So you need to do physical and chemical information evaluation for every device you're evaluating. You'll also notice that material-mediated pyrogenicity is present here, and I'll speak to that specifically in a little while. You'll notice that chronic toxicity appears, and you will also notice that genotoxicity and carcinogenicity, reproductive developmental toxicity and degradation are there at the right-hand end of the table. I've also shown for you a couple of the footnotes from the table, which is unfortunately in the document spread over 2 pages. So it's Table 1A and Table A.1b, although it's not labelled that, of course, but it's split over 2 pages, so it's quite complex to view. Where there's a G, which is the X for physical and or chemical information, that's a prerequisite information that you need for any risk assessment. And the E, that it means it's an endpoint to be evaluated in the risk assessment. And there is text there that I will leave you to consider either on these slides or when you get your copy of the document. But you can see that endpoints for evaluation is the direction that the ISO Technical Committee has taken. And this is a change for ISO to a certain extent, and it will doubtless also mean a change for regulators' guidance too. Now, moving on to some further changes, Annex B is now guidance on the conduct of the biological evaluation within a risk management process. And it's really guidance on how to conduct a risk management process for your biological evaluation. In the past, this was fairly high level and made reference in great quantity back to other documents like ISO 14971, which is the risk management for medical devices. This time, the guidance is quite explicit. It was, before becoming Annex B, a ISO technical report in its own right. There are about 8 pages, and it gives you some indication of just how seriously this is being taken by the ISO Technical Committee, that there is such a big piece of guidance about this process. It's really very informative, and it's very helpful in forming approaches to writing up biological evaluation. There are also some additional definitions for terms that are used throughout the 10993 series. There are some more categories, and particularly those categories are non-contacting, so devices that don't, in fact, contact the user or the patient. And there's also a transitory contact duration. In the past, if you had a device where you had some very limited contact, perhaps it was the power button on the machine, you would have to call that a short-term or a limited contact device because the contact time was less than 24 hours. And you had to treat it as an exposed part of the device. Now with transitory contact, that covers all of those types of applications like the power button that I've just mentioned. And it allows you to say, well, the contact is so transitory that it doesn't really require evaluation. So that's going to be very useful for a number of applications. There are also some sections on nanomaterials and absorbable and degradable materials. I've already mentioned absorbables and degradables. Nanomaterials rightly deserve the acknowledgement, and I'm sure that many of you will be aware that there is ISO 10993 Part 22, which talks about the biological evaluation of devices containing nanomaterials. And that's a very important document, and of course this document links to it. There's also the acknowledgement of ISO 18562, all the parts thereof, of which there are 4. Arthur alluded to this earlier. This is a document that was written for the biocompatibility evaluation of breathing gas pathways in healthcare applications. So it's a device-specific biocompatibility standard, and I imagine that it is the first of several, although I can't in any way predict which type of medical device will be the next one to have its own biocompatibility evaluation standard. But this is an acknowledgement that there are some devices for which there are standards specifically, and they don't necessarily fall under the umbrella of the horizontal series of ISO 10993. And as often happens in the description of changes in an ISO standard, they say there's been significant editing changes throughout the document. In other words, it's been rewritten, and heavily so. It's a much more substantial document than it has been in the past. It is now very informative and very well considered, and it's a much more comprehensive document than we have been used to in the past. So no discussion about ISO 10993 Part 1 would be complete without talking about the risk management aspect of the whole biocompatibility process. Evaluation is still, and even more so, about risk management and about risk assessment and risk evaluation and risk control. You will see the graphic on the right-hand side of your screen, which is from the guidance annex, Annex B. This graphic is actually taken from ISO 14971, and it describes the various steps in risk management. There are also other terms that crop up when we talk about risk management, because we have risk assessment, which isn't quite risk analysis, and we have risk evaluation, which isn't quite risk assessment. So there are a number of terms there, and this graphic is very helpful to bring the relationship between those terms together. I'll make the point that risk is actually something that can be considered quantitatively as the product of the severity of a harm and the probability of that harm's occurrence. And the harms associated with a medical device are in fact the biological endpoints identified in Table A.1. They are the harms that the international community has decided are appropriate for medical devices that have that sort of exposure to the body. And probabilities are able to be determined from the physical and chemical characterization information, and also, failing that, the outcomes of biological testing. If I can offer at this point some gratuitous advice, it would be that you should show safety factors where it is possible to do so. It is probably in these enlightened days unacceptable to simply say, yes, this is no problem. It would be much better for people who are presenting this information to say to their audience, this device has a safety factor in this aspect of 3.7 or 5.1 or indeed of 0.5. So show the safety factors where it is possible to do so. And a second piece of gratuitous advice would be, when considering the benefit of a device, which is part of risk management, I think that that benefit is best considered lastly, at the end. So firstly, consider the risk, and show the safety factors, and then apply the consideration of benefit. Although the expression benefit factor is often used both in risk management documents and indeed some documents in the 10993 series, it's my opinion that it is quite difficult to quantify benefit, although there are schemes published for doing so. In my belief, that benefit is more accessible to the readers of such evaluations when it is described qualitatively. But I would put that at the end of my risk management document. So they are the risk management aspects for the new edition of 10993 Part 1. There is also a considerable reliance here on toxicological risk assessment. And you'll forgive me if I find this a bit of a mouthful to say. So very often I will refer to this as TRA. And if I do so today, then I trust that you will understand exactly what my meaning is. The addition of the physical and chemical information requirement is really about chemical characterization of devices, knowing what's in a device and indeed knowing what comes out of a device to which the user may be exposed. And there is a standard for chemical characterizations, which is Part 18, and that's coming under extensive revision. And this characterization allows and indeed drives manufacturers of devices to TRA. And Part 17, which we know as allowable limits of leachable substances, is undergoing extensive revision at the moment as well. And when that's revised, it's quite likely that the title of that standard will change to Toxicological Risk Assessment of Medical Device Constituents. So having placed physical and chemical information in Table A.1, the ISO Technical Committee is acknowledging that it's something that needs to be done, and the parts that drive chemical characterization and TRA to follow on after that. And of course, the whole point of the TRA is to reduce the amount of biological testing with all of the benefits that that brings. I might speak briefly to the time frame associated with the revision of Parts 18 and 17. I can say that the documents are currently under revision. Exactly how long that will take, I am unable to say. There was a thought at the ISO Technical Committee about waiting for Parts 17 and 18 to be revised before revising Part 1, but that was overruled because it was felt more important to revise Part 1, the guidance or the ruling document for the series, and then as quickly as possible follow with the revisions of Part 17 and 18 at the technical level. So there's been a bit of an attempt to simultaneously publish revisions, which clearly hasn't worked, but I trust it won't be too long before those revisions are in place. Let me speak now to some of the specifics of the new version. One of the interesting ones is material-mediated pyrogenicity. This is a type of systemic toxicity, a very particular type of systemic toxicity that is actually considered often to be a separate thing and not a systemic toxicity, but it's a type thereof. It's a contentious issue because it is extremely rare and some would say non-existent. There is some concern that there's actually no such beast as material-mediated pyrogenicity. So I have shown there that possibly nickel salts are pyrogenic, and that may be an example of material-mediated pyrogenicity. But certainly there is biological mediated pyrogenicity, and I'm quoting there in that extensive list from the standard on material-mediated pyrogenicity. And you can see that all of the items there are biologics. They're not materials, if you like, but they're things which may ultimately end up involved in materials with devices. So in the past, material-mediated pyrogenicity probably didn't exist because materials were fairly simple. They were polyethylene or they were glass ceramic. But as materials for medical devices have become increasingly more complex, then the possibility of biologics being involved, and some of those biologics being potential pyrogens, becomes more of an issue. And this is why material-mediated pyrogenicity has risen to the attention in the 10993 series of documents. As technology changes and as devices become more diverse and more complicated, it was considered necessary to bring this to the fore as an issue for consideration. Let me speak also about FDA guidance, because really this webinar is to show how FDA guidance and ISO 10993 are coming into alignment. So the last guidance was finally issued in June of 2016. Takes a long time for guidance to be issued by FDA, and it's probably going to take a long time for them to update it, to bring it into alignment with the new version of 10993 Part 1. But there is a very strong correlation between the Table A.1 from the ISO document and the same table from the FDA's guidance document. There is a correlation on the device characterization and the endpoints for evaluation. And this is perhaps not surprising because the FDA is a fairly important body internationally. And of course, through their national representatives, they have representation on the ISO committee. And so this correlation between these is deliberate. Not that ISO has followed FDA slavishly, in no way could that be claimed, but certainly the two documents now align quite well. And FDA still maintain their X's and O's, which is somewhat confusing, but essentially any box with a mark in it in the FDA guidance table A1 needs to be addressed in the evaluation. And this quite well aligns with the ISO table A1 and the need to evaluate everything with an E in it from that table. And evaluation reports really need to be very comprehensive documents. In the past, it might have been acceptable simply to say, here are the biological tests that we've passed. That really doesn't cut it with the FDA currently, nor should it. And indeed, for the rest of the jurisdictions, it really isn't acceptable either. And the new standard edition is pointing everybody towards making comprehensive evaluation documents that are spelling out exactly why and in what way devices are biocompatible. And I should remind everybody that the FDA has some device-specific guidance documents that talk specifically to establishing biocompatibility. So, for an example, there you can see I've invoked medical gloves. There are a number of other specific devices where FDA has published guidance that speak to biocompatibility. So it may be that a manufacturer looks at FDA guidance and decides what to do about establishing biocompatibility, but then in the review process, they suddenly discover that they haven't done what FDA wanted because there's a device-specific guidance. So that's a cautionary statement there for everybody who's engaged in establishing biological safety for devices, to make sure that you understand where there is device-specific guidance, either from US FDA or from any other authority having jurisdiction. Let me show here what I believe to be best practice in terms of the outline of your evaluation document, or indeed your evaluation process. Firstly and foremost, there must be a definition of the subject device, and there are a number of things that go into that, and they're listed there in a particular order, and I'm sure that as you consider those, either today in this webinar or when you review these slides, you will understand why those things need to be very well defined and written down to be part of your evaluation. Physical characterization is an area that is perhaps not well understood for medical devices, but there are physical characteristics of devices that may influence their biocompatibility. A particular one that I will give you as an example is the way in which the surface finish of a material may change the way it interacts with biological fluids, particularly the clotting of blood. So physical characterization may be a particular issue for certain devices, which is why it should be evaluated here. This characterization is relatively straightforward and is perhaps not burdensome to achieve. Chemical characterization may not be the same as physical characterization at all. It may be a very extensive and complicated process. But chemical characterization is to facilitate the TRA. And in fact, although they're listed here as two separate items, they actually overlap and interweave with one another. So the toxicological risk assessment is actually done in and through the chemical characterization process rather than necessarily after it in sequence. Then there should be the biological data, where it exists. Not every evaluation will include biological data, particularly not biological data specifically derived on the subject device. It may rely on biological data that is existing for previous devices or is well known from published or literature. So the biological data will of course feature in this, and of course data is meaningless without interpretation, and so there should be an interpretation to follow that data. And then there should be an overall biological risk assessment which takes into consideration all of the above, but specifically the toxicological risk assessment and the interpretation of the biological data. And this is the point then where, if it is necessary, there can be some consideration of benefit, and then the evaluation would be the outcome, whether the device is biocompatible for the application to which it is intended. So those are my thoughts on the content of an evaluation outline and the process of doing an evaluation.
Thank you.
Now we have an opportunity, I think, Arthur, to field some questions from our participants.
We do. James, there's clearly an awful lot to think about here. We've got a bunch of questions been coming in during your webinar, so I'm going to start with a very simple one, actually. Dan asks a simple question. He says, hang on, what's this thing called an endpoint? The standard doesn't define endpoint. Can you just explain what you're talking about? Can you explain the concept of an endpoint?
So an endpoint would be a biological response. So let's consider cytotoxicity. So cytotoxicity is the ability to kill a cell. So a cytotoxic endpoint is, does the device actually kill cells? And it's as simple as that.
It's just the different types of
correct
effects.
That's right. It's a biological effect. It's an endpoint, and some of them are a little obscure, Dan. And I thank you for raising this point, because it's actually fairly important. For example, there's an endpoint that's called implantation, but what it really means is local effects around an implant. So when you put an implant in and you do the test for a period of time and you then take the implant out, you're looking to see, are there any local effects around the implant? And if you find them, then they are your endpoint.
Thank you. Again, on endpoints, Valeria asks, what's the new approach in the new standard when you fail just one endpoint? If you have a failure of just one endpoint from the whole list which you're required to assess, can you conduct an investigation just for that single endpoint only, maybe by retesting or doing something else, or do you have to do everything again?
Thank you, Valeria. That's a pretty important point. Let me firstly say that the ISO 10993 series of standards are horizontal. That is, they are methods to do something. They are not vertical. That is, they are not outcomes to achieve. So you are evaluating for endpoints. And when you evaluate for an endpoint and you get a result that you don't think is good enough for your device, that's the point at which I think you're saying, oh, it failed that endpoint. So if you've said, oh, I don't think it's good enough, then hopefully your characterization, particularly your chemical characterization, will have identified what substances might be present in the device that are causing that endpoint to be not good enough. And then, because this is a risk management process, you can go through risk control and you might be able to change something about the device which would decrease the probability of that endpoint actually occurring. So if you fail a biological test, if I can put it as crudely as that, which is frankly the way everyone still thinks about this, then you don't just retest it. You perform risk control. You do some change, and then you can evaluate, will that change actually improve the outcome for that endpoint? And it may not be necessary to actually test it again. I should point out right now, and very openly, that this is the way that evaluation works now, and indeed probably has always worked, but this is not necessarily what authorities having jurisdiction want. Some authorities having jurisdiction will require you to give them certain good test reports for biological endpoints. So they are simply going to require that, regardless of what you think it means. And this can be a challenge, particularly for cytotoxicity, where it is perhaps not very clinically significant, and it's a very sensitive test. It's very easy to show that you have a cytotoxic outcome, but it may mean nothing for the device. Thank you, Arthur.
Thanks, James. I've got two questions here which cover the same. I'm going to ask them at the same time. Mark says, very briefly, history of safe use. At what point, with justification, does a history of safe use suffice for physical and chemical characterization? And Tom asks that question more broadly. He says his company makes medical devices. They've been making them for 40 years. Most of the products were created prior to biocompatibility testing. They've had zero clinical issues for 40 years, but ISO is requesting in vitro biocompatibility testing on all of our products. Testing on all products is very expensive and won't provide better information on actual clinical. How do I deal with this? So for legacy products, you understand the question?
Yeah, I absolutely do.
And I question whether ISO is saying retest, or maybe, I don't know, Tom, whether assessors are asking you to retest. But anyway.
Certain reviewers may be asking that question. And this part of ISO 10993 in this new revision actually speaks to the whole point about legacy products and a history of safe use. And one of the questions that's asked in the whole characterisation piece is, has this material been used before? And so where you can demonstrate, and in FDA terms, this is called a predicate, something that has previously been shown to be safe and effective, and you can say this new one is exactly the same as the predicate, then really there is nothing more to be declared. And you can, in a literature review and using clinical evidence, you can reinforce the point that the biological evaluation of this particular device shows it to be biocompatible because we've used it for the last 40 years without it hurting anybody. So although reviewers may occasionally insist upon test results, the intent of the standard is to take advantage of everything that we know about a device and use that information wisely and well without requiring additional testing effort.
I'm going to ask a follow-on to that because I hear what Tom's saying. The material's been around 40 years. The approach taken by reviewers, and FDA are particularly firm on this, is that they want to see safety demonstrated, whether it be by risk assessment, whether it be by chemistry, or whether it be by biology or a mixture of others. They want to see safety demonstrated for the finished device,
Yes.
for the actual device as processed. And you may have had a material for 40 years. If I was to go into most manufacturers that have been around for 40 years and say, okay, show me what the material was like 35 years ago, they'd struggle to produce the records to be able to show that. So how was it made then? How was it sterilized? What were the sterile residues? All of that. So there's a bit of a caveat there, isn't there, about demonstrating safety of the actual finished device?
There is. And I think the pet expression is the final finished device. That is the device as it comes out of the package at its point of clinical application. So that would include sterilization, and it includes manufacturing process. So although a manufacturer's devices may have been made out of 316L stainless steel for the last 40 years, I imagine that there have probably been some changes in terms of the way in which the metal has been formed into the devices, the chemicals and processes that have been used, the electro-polishing methods that have been applied, the way it's been packaged, what it's been packaged in, how it's been sterilized, and what sort of shelf life it has in the sterile condition. So the final finished device is really only 1 part stainless steel and 4 parts process and process additives. And so really what you need to say is that we've been making these surgical forceps out of this grade of steel with this finishing process, and we've been packaging them like this, and we did that in 1988, and we did that in 2001, and we did that in 2012, and we're doing it in 2018 and 19, and it's all the same. And that is how you take advantage of what you have done in the past.
But to be very clear, the standard provides for that, doesn't it?
It certainly
if
does.
You've got that long record of clinical history, then that trumps the need to do any biological testing. That's the point about this rewrite, is it's saying it's a risk assessment process. Take all of the information, and you only need to do biological testing and use animals and so on if there's a gap in your knowledge.
Correct. And if you had changed the way over the years in which you had electro-polished the surface, the question is what does that actually change in terms of the surface itself? Did it mean that when you did it in the 1970s, you had a nickel oxide rich finish, and the way that you currently do it, you have a chromium oxide rich finish, well, that's a change. But if you've got the same oxide rich finish from a different process, then the outcome is the same. The final finished device is what we're interested in.
Thank you. Let's move on to another couple of questions here. I've got Elena saying, wants to go back to the idea of transient contact devices. How do you demonstrate no testing is needed? And that's not mentioned in Table A.1. Where does it appear? And is a cumulative, you need to think about cumulative contact or not?
So cumulative contact certainly is something that needs to be considered for every device, whether it's realistic to think that it's the contact will become so cumulative that it becomes short-term contact is a good question. Although it's not talked about in Table A.1, it certainly is talked about in some detail in the section of the standard that deals with contact duration. There's a particular paragraph that speaks to the transient contacting time. And interesting, it gives the example of a blood lancet or I think a hypodermic needle. So it's talking about some fairly invasive devices, but really are only in contact with just 3 or 4 seconds worth of contact with the patient, although it might be their circulating blood it's in contact with. So there is a further and full discussion about that, and I do commend that part of the document to your attention.
I think it's also making clear that really incidental contact, things like power buttons, computer keyboards, handling the outside of a monitor, that sort of stuff, is not meant to trigger a biological evaluation because the contact is so incidental and so low risk.
Yes, that's absolutely right, Arthur. People are exposed to the contents of their motor car to a far greater extent than they are to most medical devices.
Thank you. Ian just says, how much and to what level is chemical characterization required? I might add to that question to clarify. We're not talking about having to go off and do an analysis every time, are we?
So, Ian, thank you for the question. Chemical characterization is like a piece of string. No one knows quite how long it is until you get to the end of it. In Part 18, there will be guidance about the extent of characterization. I've seen the flowcharts that go with this discussion, which will be part of Part 18. And 2 things to point out. The first thing is that Part 18 is pretty clear about just doing the chemical characterization that you need to do. You don't have to do everything. You just do what you need to do to show what it is you're trying to show. So that's the first thing. And the second thing is that in terms of the flowcharts, they are flowcharts with multiple entry and exit points. So you don't start at the top and work your way through $3 million worth of analytical chemistry, for example. You start and you do as much as you need to do, and then you get out. So you might get away with $300 worth of characterization. If you can do it all by getting the information from your material manufacturers, then that's what you do. And so on Table A.1, that particular column doesn't say characterization. It says physical and chemical information. In other words, get some information. And how you get that will be very dependent on the context of what you're doing. As I always say about biocompatibility, context is king.
Thank you. And let's just put some context there. So if you're dealing with an implantable, biodegradable device, which is a complex polymer, then you're going to be needing to do some chemistry, aren't you? You're going to have to look at what comes out, what degradation products are. That's going to be a substantial investment in understanding the chemistry. Whereas if you're talking about that piece of 316L steel, the only thing you're really going to be worried about there is that you've got a certificate of analysis from the manufacturer that says it is indeed 316L steel, and that you make sure that your process cleans it thoroughly and you know there's nothing left on the surface. You might need to do something that looks at to confirm there's no processing aids or sterilant residues left on the surface.
Yes, that's right. They are very different approaches, and it does absolutely depend on what the device is made of and what that categorization is. And as you rightly point out, Arthur, something that is implantable is going to require a lot more scrutiny than something that is, a surface contacting device. And of course, although the ISO standards do not deal in the classification of devices, we all know that authorities having jurisdiction, have devices that are classes 1, 2, 2A and 3 usually. And the more invasive and the more serious a medical device is, then the higher its classification. And I think we should rightly expect that the higher classes of classification for devices will require a more thorough treatment in terms of biological evaluation.
Although that piece of steel is a class 3 device if it's an orthopedic implant. And it's
yes,
an implant.
It certainly is.
So, it depends on the context, as you say.
So if it were a class 1 piece of steel, you might just show that your process flow does all the right things to make it clean enough for what it does. But if it's an implant, then you probably need to show that your process flow makes it clean enough to be an implant, and then you characterize the residues from the process to show that it's clean enough to be an implant.
Okay, a really technical question one now. James says, if a material directly causes the production of biological mediators of pyrogenicity, do you consider that material-mediated or biologically mediated? And he gives an example. An extract from an orthopedic implant interacts with bone osteoblasts and causes interleukin-6 production and secretion. Is that materially mediated or biologically mediated? And maybe I'll ask the supplement, does it matter? It's the effect that matters, isn't it?
It is. So it doesn't matter because the end point is observed. There will be some pyrogenicity because the interaction between the device and the body causes a particular outcome. This is almost the textbook definition of biocompatibility, that the interaction between device and body doesn't cause an adverse biological response. So I would call that material-mediated pyrogenicity because the material drove the outcome.
Indeed. Thank you. Alan asks, I see a lot of labs offering a service called extractables and leaches. And what is that? And do you need it? Does he need it? How does it help? Yeah, extractables, leachables. Do you want to just go there and explain
Certainly.
that?
Extractables and leachables speaks actually to chemical characterization. Extractables are things that under very exaggerated conditions can be dragged or ripped out of devices and materials, whereas leachables are the substances that come out under clinically significant conditions. What really happens when you use a device, in other words, that's what a leachable is. Labs are offering this service because it's a precursor to toxicological risk assessment. This is chemical characterization. I think many labs are just offering this as a service because they're jumping on the bandwagon. I suspect that manufacturers don't currently know what they need or indeed how to do characterization. I think it's probably a little preemptive of many laboratories to be offering this service because it's probably a revenue-raising issue without actually adding a lot of value to the marketplace.
It's an area that's under very active review in the revision of Part 18.
Correct. Yes.
And the question really is which of these approaches is right, and in some cases neither. You don't need to do it. In some cases you just do extractables and some you just do leachables. The historical approach has been to start with extractables in most cases, use aggressive solvents, use raised temperatures, because if you can show from a study like that that nothing dangerous comes out, then you've got a huge margin of safety. Yeah. But with increasingly complex designs and increasingly advanced materials, things designed to degrade or whatever, that approach isn't always appropriate, is it?
No, it certainly isn't, Arthur. I think there's a hierarchy of chemical characterization, and at the top of the hierarchy would be the formulation. That is, what is the list of all of the substances that go into a device? So that's what goes in. Extractables are then what goes out. So it's a kind of a way of reverse engineering, if you like, the formulation, if you don't have that. And then leachables would, I think, almost always be a subset of extractables, which are clinically available. But sometimes it's quite difficult to produce the clinically available ones because they only become clinically available when they're implanted or something like that. So you do extractables or leachables, or indeed get the formulation according to how much information you need for your particular device. So again, this speaks to the point that I made from part 18, which is you do as much as you need in terms of chemical characterization. Certainly, extractables and leachables can be very valuable, and I wouldn't want to undersell that. I think all manufacturers, though, would be familiar with the issue of getting a lab to do what the manufacturer needs them to do rather than what the lab wants to do.
One very last quick question, because we're almost out of time. We're coming to the top of the hour. Carl asks, when's part 17 and 18 going to be ready?
Oh, Carl, I hope that part 17 and 18 will be published in 2019. So that's next year. I know that there's a very heavy amount of work going on for both of those parts, but I think that those parts will probably be able to be published in the next year or so.
Thank you. So I think that brings us to an end. I hope this has been a valuable presentation. James, I always learn some more when I hear you speak on the subject. As you can know, James is a very knowledgeable expert in this field. We'll be back next month. In the meantime, you can follow us on social media, on Twitter and LinkedIn, and also view this webinar and others in the series on our YouTube channel, or go to our website where you